Good afternoon, everybody. Today we're doing chapter 12. I'm not expecting a lot of folks to join in, but if they do, that would be great. I'm going to go ahead and get started, assuming that there's not going to be very many people. So today we're doing chapter 12. I'm going to go ahead and start that. Okay. Today we are talking about neurons and how neurons work. Okay. Different parts of a neuron. The cell body is the area that you see up there near the top. It's the side of the nucleus that just below where it says cell body soma. And the side of neurotransmitter synthesis. We're going to be talking a little bit about what are neurotransmitters and what are they for. Dendrites, as you can see in the far left, upper left, are always bringing signals in towards the cell body. That's incoming signals towards the neuron, senses, for example, or connection with other neurons. The axon is the large structure that you see heading off to the right and downward. You can see it's covered with myelin sheath. We're going to be talking about the significance of that in a bit too. By the way, this figure is in your text if you want to look it up. The neuron or myelin sheath, what it does is it insulates the axon. The way that it works is very much like the plastic around a wire. So what is the plastic around a wire for? Well, it's partly, yes, to keep you from getting electrocuted, but it's mostly to keep the electricity from going into the ground and to maintain the signal through the wire. And that's really the same thing that's happening here. We're going to be talking about the nodes of Ranvier later on. And it also allows it to speed up because jumping is faster than burning. And I'll explain what I mean by that later. Okay, talking about different kinds of neurons. There are afferent neurons. Afferent neurons are sensory neurons. Those are the neurons that bring in sensory information from our environment towards our central nervous system. The first type is called a pseudo-unipolar. What that means is pseudo means false. Unipolar means that it seems like only one pole, but it actually has an axon and a dendrite fused together. But the action potential may bypass the cell body. We're not going to talk much about those. In bipolar, as you guys know, bipolar means having two ends, one axon and one dendrite of roughly equal size with the cell body in between. And afferent neurons, that's what we were just finishing. And then now we're talking about interneurons, which are neurons of the CNS. The interneurons means that they are neither sensory nor motor, but they're basically helping you think and make decisions about your environment. So the first type of interneuron is called an anaxonic, which means that it doesn't have an obvious axon. It may have an axon, but it's not obvious. In multipolar, it's highly branched. This is the most common one found in the brain. Lots of connections between different neurons. Efferent neurons are the third type. These are motor neurons. These are the neurons that send signals from your central nervous system or other similar structure out to your muscles or glands. It's traditional multipolar neuron shape. There are many dendrites with one axon. I showed you a picture. Those are the ones that we'll be spending most of the time talking about today. Okay, you can see unipolar is there, bipolar upper right, and multipolar is down below. That's the most common shape. Okay, metabolism and synthesis in a neuron. The cell body is the site of ATP generation. We talked about how that works. There would be lots of mitochondria there, and neurotransmitter synthesis. Question, why do neurotransmitters have to be synthesized at the cell body? It's because that is where the DNA is, and DNA is going to make a piece of messenger RNA and therefore use that messenger RNA to generate the neurotransmitter, especially if it's protein-based. Neurotransmitters then, when they're created, the cell body are going to move along the axon away from the cell body down towards the synapse. The synapse is the gap between the end of the axon and the next cell, which is either another neuron, a muscle cell, or another kind of cell altogether. The synapse is the narrow gap between the axon and its target organ or other neuron. Now, there's two different ways that neurons can communicate with other cells. The first one is through a synapse. The other one is through, which is called a chemical synapse. We'll spend a little bit of time talking about that. Now, the main neurotransmitter that we're going to be discussing today is called acetylcholine, abbreviated ACH. So make a note, ACH is acetylcholine. Other neurotransmitters, you may have heard of dopamine and serotonin. Those are neurotransmitters that can also work in the brain. They're released from vesicles. Vesicles are little bubbles that contain chemicals in it. And They're found near the presynaptic membrane. I'll show you how that works. It means that these little bubbles full of these neurotransmitters travel to the end of the axon and wait for a signal to dump their chemicals into the gap and send a signal to the next cell, especially a neuron. The process by which it dumps those chemicals is called exocytosis. So then when the chemicals are dumped through exocytosis into the gap or synapse, they diffuse across the gap and then stick to receptors on the postsynaptic cell membrane. So on the next cell, there's going to be receptors there that bind those neurotransmitters. And when the neurotransmitters bind, it's going to cause a chemical reaction in the cell, usually the initiation of a graded potential or another action potential. I'll explain what those are in a bit. Okay, the NT or neurotransmitters cause a reaction on the postsynaptic membrane, such as the continuation of an action potential, or at least the generation of a graded potential, which I'll explain, or a muscle to contract, which is also based on an action potential, but slightly different type than you would see in a neuron. Okay, so we're going to be talking about cells that are called glial cells. Now, there are two different kinds of cells in the nervous system: neurons, which we're familiar with, and glial cells, or neuroglia. Now, glial cells are kind of helper cells. They don't do any actual sending of signals, but they help maintain the integrity and help the metabolic needs of the neurons. So they don't really action potentials, so they're not going to be sending signals. But they support and nourish the neuron cell bodies, especially in the central nervous system. We're going to see there are different types of neuroglial cells in the CNS versus the PNS, central nervous system versus peripheral nervous system. They also help remove waste from neurons, so they really are a way to help the neurons move things into the blood and from the blood. They also form myelin sheaths around axons. I showed you that earlier. That was that whitish blue material that goes around the axons, insulating them, helping the signal move faster. Now, in the peripheral nervous system, the cells that do this myelin sheath formation are called Schwann cells. And I like to think of them as kind of ice cream cells. Have you guys ever heard of Schwann ice cream? They wrap around kind of like a cinnamon roll. Yes, my mind is often on food. And they are insulating it in the peripheral nervous system. Now, in the central nervous system, our brain and spinal cord, we use cells that are called oligodendrocytes. Big word. You can break it up or understand it by breaking it into small bits. Oligodendrocytes. Okay, and ependymal cells are also found there. They form barriers between fluid compartments, such as the blood-brain barrier, which is there to prevent chemicals from your blood from getting into your brain that aren't supposed to be there. Microglia are other types of glial cells. They are scavenger, kind of white blood cells to a certain extent. They defend and give metabolic assistance to the neurons. Okay, so you can see these are the glial cells. This is the image directly from your text. You can see the oligodendrocytes are there, the microglial cells, the astrocytes, which I didn't mention, which look like stars, and then the ependymal cells you can see separating the blood from the inner part of the central nervous system. Okay, now I did mention what a graded potential is. Now, what I want you to think of is a graded potential is kind of something that can go on a scale, a different level of intensity. So, for example, I'll give you my tried and true analogy. There's certain amounts of pressure that you could put on a toilet handle, right? You can touch it very lightly, you can touch it a medium amount, or you can push it very hard. Generally, only pushing it very hard will make the toilet flush, but there's different amounts of pressure. The analogy I'm going to use for graded potentials is how hard you would press on the handle of a toilet. Okay, so what it means is there's signals that are coming into a neuron from other neurons or other sensory structures that are starting to destabilize it and open sodium gates and allowing sodium to come in, and that's causing what is called a graded potential, start to rise up. It varies in strength. It can be a little bit, a medium, or a lot. And what we're going to see with action potentials, there are no varying strengths. There's either on or off. So in this case, graded means different levels, as in your grades, different grades that you can get. In contrast, you can't really normally get half of a toilet flush. You can't normally half fire a bullet. So the way that a bullet flies out of a gun and the way that a toilet flushes is very much how an action potential works. But in this case, it's how much pressure you put on the trigger or how much pressure you put on the handle that's going to determine, do you get that effect that's coming later? So it loses strength over distance. It can fade in the same way that waves on a lake or pond fade out in their strength. We're going to see that's a pretty good analogy for how these signals spread. They move much more slowly because they don't jump, and I'll tell you why action potentials are so much faster, and we'll see it. A good analogy for that is do kangaroos walk faster or jump faster? Well, obviously they jump faster. So we're going to see that action potentials move kind of like kangaroos. But in this case, the graded potential are slow. They don't jump in the same way that action potentials do. Okay, so what they're going to do is once this signal starts spreading through the cell body, which is generally where it happens, it travels to a place called the trigger zone. The anatomical name for it is the axon hillock. Now it gets to the trigger zone. If it's strong enough, it'll generate an action potential, which is the toilet flushing or the firing of the gun. So if it's not strong enough, it's called subthreshold, means it's below the threshold at which an action potential will happen. So it's too weak, and it started weakening over time, and by the time it got to the trigger zone, or axon hillock, it wasn't strong enough to cause this chain reaction called an action potential. In a suprathreshold, that's above the level needed to generate an action potential, it does cause an action potential to form. So as you can see, this is the graded potential, this reddish colored material. This image is not from your text, it's from another text. But you can see as you move away from the stimulus, which is sodium coming in as a result of stimulus, which is action potentials from an adjacent neuron, you can see the sodium coming in starts to generate a graded potential, and you can see it starts to fade away. If it's not strong enough, it can't make it all the way to the other end of the cell. Okay, so if you were to measure this, and you were at a sub-threshold graded potential, you can see right up where it starts, right at the stimulus source, it is strong enough. But since we said it fades away right in the middle, it's just barely enough to have it. But the most important thing is where it is at the trigger zone, axon hillock. Notice that down here, the signal is too weak, so it just disappears. It doesn't get propagated down in the same way a fire is propagated. In this case, if you add a little bit of heat to some fuel, it's not going to catch on fire, and it's just going to cool off. Now let's look at the other one. This is a suprathreshold. What if the signal is much, much stronger? We see the strong signal here, and it does start to diminish, but not so much as to prevent this chain reaction. And you can see it looks like a lightning bolt. And the reason why they use a lightning bolt is it is an electrochemical reaction, chain reaction, that functions very much like a brush fire. And I'll be using that analogy of a brush fire. Now I have lots of analogies here. I have guns, I have toilets, and I have brush fires. They all kind of work if you understand how they work. So in this case, the graded potential was suprathreshold when it reached the trigger zone, so therefore an action potential was generated. Okay, what happens to the membrane during action potential? And this is one of the essays that you guys are going to have from this chapter, so you do want to pay really good attention. Pause this video, rewind it, and take more notes. Remember, I don't want anything from Yahoo Answers or from, medical websites. I want either from the book or from this lecture. So pay attention and take some good notes. Okay, so what is a polarized cell? Now we talked before about how the sodium-potassium pump works, and you guys just finished a test on how the sodium-potassium pump works. What it means is that sodium has been pushed to one side of the membrane and potassium on the other. Now there's a big gradient on both sides. And it means then if you open those gates, that big amount of sodium or the big amount of potassium, that difference in concentration, the concentration gradient, well, if you open those gates, it can go rushing through. And that's really what's going to happen during an action potential. Okay, so the sodium-potassium pumps have readied the membrane for the action potential by pushing sodium to one side, potassium to the other. There's high levels of sodium outside the neuron, low inside. If your notes don't say this, please fix it. What's in this PowerPoint is correct, not what is in your notes, if there's any divergence. Okay, so there's low levels of potassium outside, but higher inside. So high potassium inside, high sodium outside, and they're separated. And this is a perfect way to, it's kind of like how a toilet bowl is full of water. When you fill a toilet bowl full of water, you can open the flapper and the water will go rushing through the toilet bowl. So, but it's only going to work properly if you fill the toilet bowl all the way or most of the way up. Okay, so we saw that in one of the previous slides, if a graded potential with enough strength arrives at the axon hillock or trigger zone, it's going to cause this chain reaction to happen. And what is that? It's going to be a whole bunch of sodium gates going to open. And that stimulates the action potential to begin. So what happens at the beginning of the action potential? Well, the very first part is the increase in graded potential to threshold, and then the next step is called depolarization. Now, when those sodium gates start to open, the whole bunch of sodium comes rapidly diffusing into the neuron. That's going to cause the inside of the neuron to become very positive, because as you can see, sodium has a positive charge. So if a whole bunch of sodium ions are coming in, the inside, instead of being negative 70, which it is at the beginning of the polarized membrane resting membrane potential, it goes much, much higher. We'll see that in the figure. It's going to go from negative 70 way up to 100 because of all the sodium coming in. At the signal peak, when all the sodium has come in, at that point only, after the sodium has come all the way in, then the potassium gates open. So one of your review questions says, do potassium gates always open after sodium gates? And the answer is yes. Sodium has to complete its job. As soon as it's finished this job, then the potassium gates open. Now, the idea here is that sodium came in, making the inside very, very positive. But what happens when the potassium goes out? Well, it's going to go back to negative again. So it comes in, it goes up, potassium goes out, goes down. So it's like a little blip, which is information, which is your action potential. It's always 100 millivolts in amplitude. And this is independent of the graded potential strength. Now, this is another review question. So the question I have is, for you, just using common sense. If I press a toilet handle just barely enough to make it flush, with just enough force to make it flush, is that going to flush any better or worse than if I hit the toilet handle really hard? Will that make a better flush? Answer is no. As long as it's enough to lift the flapper, it doesn't matter how hard you hit it, whether just barely enough or really hard. You still get the same flush. Whether you pull a trigger on a gun really, really hard or just barely enough, the bullet's still going to fly out of the gun. So the idea here is it doesn't matter how intense the graded potential is. The action potential is always 100 millivolts. It doesn't matter how much the graded potential is. So try to remember that. Okay? In repolarization or hyperpolarization, we're going to see that later on. The potassium now is fully diffused out, so it went all the way up and all the way down, and it's going to actually be a little bit lower than it was at the resting membrane potential. And here we are. This is the figure that's going to be on your test, and I'm going to ask you to explain. There's another figure coming up that will give you a little bit more detail on what's happening at these different stages. But for now, just pay attention and take notes. Okay, step one, at rest. This is before the arrival of a graded potential. Sodium-potassium pumps have pumped sodium-potassium to opposite sides. You can see it's at negative 70 millivolts. This has partially due to the leakage of potassium, but a whole lot of factors that aren't important at this moment. Now, at step two, the stimulus is applied. This is the destabilization of the cell membrane near the cell body. What's going to happen now is a little bit of sodium is going to start to come in as a result of the stimulus. Now, what we're assuming here is that the graded potential, you see the graded potential is rising here. This is the rising of the graded potential. And as soon as it hits threshold, it turns from a graded potential into an action potential. So everything from this dotted line all the way to the top is voltage rising as a result of sodium ions coming in. So please note, voltage rises here rapidly because it's all those sodium ions coming in that have been pumped to the other side of the membrane, especially in the axon. Now, this rise in sodium has to do with sodium coming in at the cell body. This big rise is what is happening at the cell axon hillock, the beginning of the axon. Now, the falling voltage has to do with the potassium going the opposite direction. So all the sodium was coming in, making it rise. Potassium goes out, now it drops because potassium is also positively charged. If positivity leaves, you have negative. Notice it overshoots a little bit. You have the end of the action potential, and then in step 6, return to rest means the sodium-potassium pumps are going to start doing their thing and getting you back to the resting membrane potential. But here's the point. During this actual drop-off here, when the potassium is going out, you can't have another action potential because it's like saying, when's the only time that you can't flush a toilet? Well, when it's in the middle of a flush. You can't initiate a new flush while it's in the middle of a flush. As a matter of fact, you can only really get a good flush until after it has started to fill up a significant amount. And that's way over here past step 5. So anywhere from 2 all the way to 5 is what is called the refractory period. The refractory period means it cannot have another axe potential while it's waiting to reset. Okay, this is the figure that's going to be on your test, but I'm going to give you a couple more figures that will help you understand some more about what's happening here. Okay, positive feedback loop. Now, we talked about how negative feedback loops are really important for homeostasis, which means keeping things just where they need to be. But in some situations, you want to get away from negative feedback, away from homeostasis, because you're trying to do something which is unusual. In this case, instead of keeping a neuron nice and steady, the sodium-potassium pump, you're going to get away from that. A whole bunch of sodium in, a whole bunch of potassium out. It's a positive feedback loop. And then what that means is the beginning of the axe potential actually causes that reaction to spread down the length of the axon. So instead of getting back to normal, it actually causes everything next to it to go. And that's very much how a wildfire works, is that the fire in one spot burns, heats up the adjacent area, that catches on fire, that heats up the area next to it, and it just spreads. So the axe potential is going to spread very much like a wildfire or a brush fire, because what's happening here is going to trigger what's happening right next to it. And it goes until it's completely finished. Now, I mentioned refractory period. The first kind of refractory period is called the absolute refractory period. It means there's nothing at all you can do to make the axon or neuron have another axe potential. So sodium channels are resetting their membrane potentials. That really hasn't had a chance to pump sodium potassium across the membrane. No second axe potential may be initiated during this time. This is that whole up and down portion of it. And what this causes is it makes sure that the signal always goes from the cell body down the axon and never the opposite way, because of the fact there is this absolute refractory period. So you can't have the signal spreading back up the neuron towards the cell body. It always goes from dendrites to cell body to axon terminus and to the synapse. It always goes that one way. In relative refractory period, it's kind of like when the toilet is halfway or 3/4 of the way full. You can flush it again and get a fairly decent flush if you want. But where this diverges from the analogy is that if you have the only way that you can get it to go during the relative refractory period, that's when it's starting to reset. There's some sodium potassium on opposite sides. You need a really strong gradient potential to get another action potential. Whereas in a flush, you don't need a stronger handle push. But in this case, you need a strong gradient potential. You can get an action potential happening, but you haven't completely moved the sodium potassium to the opposite side. So there's relative and absolute refractory period. Okay, this is the figure that I was telling you about. I love this figure, and this is what you should use to give you the information that you need to write your essay. Even though this isn't in your text, this is a terrific figure that I found elsewhere. The reason why I like it so much is it shows three main things. The first is what is happening to the sodium-potassium gates, which is this blue and yellow channel going from left to right. It's showing you what's happening with the sodium-potassium gates all along the way. This is a terrific way to get more detail into your essay. Now, please note the axe potential here is red, but what is happening to the sodium is in yellow, and what's happening to the potassium is purple. So they're showing each one separately. So it shows you the sodium coming in, and then it shows you the potassium going out. This is great because it also shows, hey, this is what's happening in each of these parts. Sodium moving here, potassium moving here, resetting here. And my favorite part of this is excitability. And what does excitability mean? It means how likely is it to be able to generate a new action potential. So here at the beginning, while it's ready, the excitability or the ability to generate an action potential is very high. Then when you start this, the beginning of the action potential goes to 0. That means that is absolute refractory period. It doesn't matter. There's nothing you can do to make another action potential. Now, as we go from 2 milliseconds onward, you can see there's increasing excitability. And what this means is the higher the excitability, the less of a gradient potential it takes to trigger another action potential. So it takes a really strong gradient potential right here to make it go off. And by the time you get back to the beginning, you have a normal amount of gradient potential needed to make an action potential. If I were you, I would use this figure to answer your essay and not the one I provided. The one in your text is kind of weak, doesn't have a lot of information in it. This one has much more information. I'd be happy to explain it more to you in lab. I guess that's tomorrow's lab. Okay, there you go. Okay, what are the different stages of an action potential? So here's the actual notes. Okay, this is another review question. Action potentials are all or none. Action potentials cannot be partial. It's almost like saying, I'm kind of pregnant. Well, you're either pregnant or you're not. The bullet either fired out of the gun or it didn't, and the toilet either flushed or it didn't. Now, we're discounting those kind of, aborted flushes that happen once in 100 or once in 1,000 times. When you flush the toilet, it flushes, and if you don't, you don't. It doesn't. Okay, so axe potentials cannot be partial. Can't be a half axe potential. It's either going to go completely or not at all. The signal does not diminish over distance, as in graded potentials. In the same way that a wildfire with fuel is not going to get any cooler, the axe potential is going to continue to generate the signal all the way down the axon because it's going to get re-stimulated, re-strengthened by the sodium gates sitting right next to it, to the opening, adding more sodium. So the action potential spreads throughout the entire neuron, but it doesn't matter. The only place that matters is down the axon. Now, this is where it gets a little bit tricky. You probably think it's already tricky, but here's the deal, and it's not easy, so please pay attention. The intensity of the stimulus that originally occurs has nothing to do with the intensity of the action potential, because I said it's always 100 millivolts, and it doesn't ever change in intensity. It's always either on or off, like a 1 or a 0. But the intensity of the stimulus above threshold, whether it's a little bit of stimulus that makes something happen or a really big stimulus, is coded for by the frequency of action potentials. And let me explain what that means. If a signal goes blip, and then it resets and goes blip, each one of those being an action potential, blip, it means the signal is just barely strong enough to generate action potentials. But what if the stimulus is really, really intense, or really hard touch, or a really intense flavor? It would be more like blip, 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 faster, more frequent action potentials tells the brain, hey, this is a greater, more intense stimulus than if fewer ones. So it's kind of like Morse code. The more that come per second, that codes for the intensity of how strong the initial one was. So be sure to put that when I ask you about it. So frequency, it means the number of APs or action potentials per second. Higher frequency means it was a more intense stimulus. Lower frequency, fewer per second, a less intense stimulus. So if it's a high frequency, it was a strong stimulus, and a low frequency means it was due to a weak stimulus. It's strong enough to generate action potential, but not strong enough to generate lots of them very quickly. Okay, so this is what's happening when you get suprathreshold. This is right in the axon hillock, and this is what's happening here. A graded potential that's above threshold reaches the trigger zone. What happens is, if it's strong enough, sodium gates start to open. Now, what makes this interesting is when these start to open, they're going to cause a chain reaction right next to it. Notice that there's nothing to the left here. It's only to the right. This is heading down the axon. So when the sodium comes in, it's going to trigger the ones next to it. So notice sodium is coming in both directions, but because there's no sodium channels here that are going to respond to these sodium ions, the ones to our right, down the axon, are the ones that are going to respond. So as the sodium comes in, they travel over, and they're going to cause these ones to open. When this sodium comes in, it goes over this way, causes these ones to open, kind of like a forest fire, wildfire. And that's going to spread all the way down because the inrush of sodium is going to cause adjacent sodium gates to open. You can see now, after the sodium has come in, now the potassium is going to go out. So this is anatomically what's happening. Even though the movement of sodium is happening first, then the potassium happens second. And so it spreads down this way, and then the potassium is going to go out. The refractory region means that this is where the potassium going out and it's resetting. No action potential can be initiated here. Okay, so speed of conduction. What makes an action potential really fast? Well, I mentioned kangaroos, right? If you ever see a kangaroo walking, they walk fairly slowly. If you ever see a kangaroo jumping, they move really fast. Why? Because they're jumping from one section of the ground to the other and flying through the air between their jumps, and that's far faster than walking. So here's another thing that you should know. If axons that have a large diameter, it means thicker, when you look at them in cross-section, they're going to allow action potentials to go more quickly because more sodium can flow through it in the same way more water goes through a pipe that's larger than smaller. The other thing is myelination. That's that wrapping of this fatty material, this bluish-white material that I told you was oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. When there's myelination on it, it causes faster conduction because the signal jumps from node to node rather than spreading like a fire. Now, fires can spread really fast, but think about if a ember flew from the fire through the wind and landed further away. It could start a brush fire there. That's far faster than a fire would spread if all was happening was no wind and it was just burning and burning and burning and burning. It would take a while to get all the way across, no wind. But if an ember flew from one part of the fire into another part, you could make the fire spread far faster because it's jumping, and that's what's happening from node to node, or node of Ranvier to node. So the myelin in those spots between the nodes prevents the sodium-potassium leakage and loss of the electrical signal out of the cytoplasm. What's the significance of that? I'll show you. This process of jumping from node to node is called salutatory conduction. The action potentials appear to jump from one node of Ranvier to the next because they come in, they spread very quickly under the myelin, and then they cause another chain reaction, and then they spread. It turns out that diffusion down the axon is far, far faster than the chain reaction of opening sodium-potassium gates. I'll show you. Now, you guys may have heard of multiple sclerosis, the inability to move. What happens here is that the myelin is damaged or destroyed. And what happens is the sodium-potassium start to leak, and you lose the signal in the same way that if you put bare wire on the ground, the amount of electricity getting to the end would be diminished because it would go out into the ground. The other thing that can stop this is chemicals that block ion channels. For example, certain neurotoxins from venomous or poisonous animals like snakes or scorpions or whatever. And cane drugs. You guys have heard of Novocaine and lidocaine, and even cocaine, believe it or not, can numb your gums if you put it on there. So cane drugs, they block the ion channels, prevent the signal from going through. That's very handy if you're at the dentist's office. They inject Novocaine in there. It blocks the signal. It also blocks motor control, so you can't move your mouth very well, but it blocks the pain signal. So until that diffuses away into the blood, the nerve doesn't work, but it's temporary, so that's good. Okay, so let's talk about how an action potential spreads through a normal neuron. Now notice we've got a signal coming through here, and it's got enough strength because it hasn't leaked out right here where the myelin is. So it spreads very quickly. There's a little bit of loss in strength, but when it gets to another node, it gets recharged by the fact that it opens these sodium gates here. So notice it gets boosted. It slows down a little bit through, but it's getting boosted by more sodium ions. Now, the next node, it's going to shoot under the myelin, losing a little bit of intensity, but the speed is really fast under there. And when it gets to another node, it gets recharged, and it keeps doing this. So it's speed, gets recharged, speeds, recharged, and then it makes it all the way to the end. Now, in multiple sclerosis or MS, notice that the signal is diminishing, but up here it doesn't diminish very much. It diminishes a little because of the insulation. Now, in this case, because the myelin sheath is not there or diminished, the signal leaks out, and so this chemical signal gets so low that it may not trigger another recharge of the signal. So in multiple sclerosis, the signal can stop because it doesn't have enough. And notice that there's no sodium and potassium gates here because that's not how it's built. So if you have myelin, it has enough intensity to get to the next node where it gets recharged and speeds on. Here, it loses intensity quickly because the signal leaks out into the tissues, and it may not have another signal make it to the next one. That's why people with multiple sclerosis often have difficulty moving parts of their body because their neurons are not working. I hope that makes sense. Okay, the synapse. Okay, now when we get to the end of an axon, we're going to talk about what happens at a synapse. A synapse means a gap between the end of the axon and the next cell. Now, the first kind that we're going to be talking about are called electrical synapses. Now, electrical synapses are very different than the ones we're going to be talking about later on. These are called gap junctions. Gap junctions are basically two cells are physically next to each other. When the signal arrives at one, it just automatically spreads to the next. There's a physical connection between the cells. There's no gap like we see in a regular chemical synapse. The very fast conduction of action potentials, which you would think, isn't that great? The problem is there's no way to regulate. Once the signal comes through, it's automatically going to go to the next cell. There's no way to slow it down. In a chemical synapse, the body can put either speed up or slow down chemicals in there to either enhance or slow down the speed of the signal, and that's something the body really wants to do. So for example, in the cardiac muscle, your heart, all of those muscle cells are electrically connected. So the idea is you always want the heart to completely contract. You don't want it to partly contract, and you don't want some of them to contract and not others, because otherwise your heart wouldn't pump the blood that you need it to. So that's a good time to have electrical synapses. Now, chemical synapses is what we're going to spend most of our rest of the lecture talking about. There is no physical contact, so there's a little bit of a gap there. And that's where the chemicals have to diffuse through. So the presynaptic terminal, what does that mean? Pre means before the gap. The terminal means that's the end of the axon where the signal that we were just talking about is going to end up. So neurotransmitters were synthesized up at the cell body and transported down to the axon terminus, or right before the synapse. And they're waiting for a signal to dump their chemicals. So the neurotransmitters were synthesized at the cell body. They're waiting at the terminus to be dumped. They're stored in vesicles, which are little bubbles full of these acetylcholine or other neurotransmitters. Now, the key is calcium is needed here to allow these bubbles to stick to the membrane and dump their chemicals. So calcium is important. This is one of the ways that we talk about why calcium is important in other places than just your bones and teeth, because they're actually important in terms of having neuron signals go from one cell to another. And we're also going to see that they come up as very important in muscle cells, and that's later. And once those calcium ions allow the vesicles to bind to the postsynaptic or presynaptic membrane, it's going to dump those neurotransmitters. And what they're going to do is diffuse through the cleft or through that little gap and stick to receptors on the postsynaptic cell, which is the second cell. So the neurotransmitter receptors are going to grab hold of those neurotransmitters. They're going to start opening sodium channels, and you're going to start generating another graded potential. So that's how the signal goes from one cell to the next. You're going to see an increase in the graded potential, eventually another AX potential, and that's how the signal spreads from one cell to the next. Okay, this is a figure from your book. But what I'm going to ask you to tell me about is what happens, what are the steps that happen? It's not shown really clearly here. So let me walk you through it. Okay, so we have the presynaptic neuron. You can see the synaptic vessels are containing acetylcholine, but what happens is when the AX potential comes down here and arrives all the way at the bottom, what that's going to do is going to allow calcium to come inside and allow these vesicles to dump. So when the calcium comes in and binds to these, it causes it to stick to this membrane and dump its chemicals. You can see the chemicals are starting to diffuse across this little tiny gap. It's exaggerated here. It's very, very small. But you can see these ligand-gated channels with receptors for neurotransmitters. What's going to happen is these are sodium channels. When these acetylcholine particles bind here, it's going to open the sodium gates and allow more sodium to come in. That's a graded potential in this cell. So the more acetylcholine comes in, the more sodium gates open, the greater the graded potential is in this cell. And if it gets high enough, bingo, you got another AX potential. But that's what's happening there. So these are the different steps. So arrival of the AX potential, movement of calcium, which this one does not show. The calcium allows these vesicles to bind to this membrane, causing the acetylcholine to diffuse. It binds to these ligand-gated channels. They open, allowing sodium to come in. That generates a graded potential, potentially high enough to make an AX potential in the postsynaptic neuron. Hope that makes sense. Ask me about it tomorrow in lecture, or in lab, excuse me. Okay, so this is another image showing you this. This is the one that I would use if I were you. This is not in your book. But this shows the actual steps. So there's your AX potential coming down the axon. It comes around here, and as soon as it hits these voltage-gated calcium channels, it causes that to open. The calcium comes in. The calcium connects these synaptic vessels that I showed you in the previous figure with the docking protein. The docking protein tears open the vesicle. It causes it to diffuse across, binds to the receptor, and as we saw in the previous figure, that opens sodium, the channel for sodium ions to come through, and now you get a graded potential here that could potentially. So this kind of clarifies what I was explaining in the previous figure. Okay, Now, once the neurotransmitters are there, and by the way, I will be asking you, how does the body get rid of these neurotransmitters as part of your essay? So please note that it's not just the acetylcholine and opening up sodium gates, but how does the body get rid of them? That's part of the essay. So pay attention. So one way that you can get rid of them is you can recycle them back into the presynaptic neuron for reuse. You can just suck them back up and put them in a vesicle again, and you're good to go. If they didn't stick to any of the receptors, you can just recycle them. It's the easiest way. The other way you can do it is you can break it down into the two components. Now, acetylcholine is made up of acetic acid and choline, which are two separate chemicals. So if the enzyme breaks those up, those won't stick to anything and cause a chemical reaction. So those will either make it into the blood or be recycled back up to the cell body and turned back into acetylcholine or other neurotransmitters. The last way is instead of breaking it up or recycling it, it just goes into the blood and leaves the area. So these are three different ways that your body inactivates or eliminates the acetylcholine or other neurotransmitters. So that's the end of the essay. Don't forget to talk about this in your essay. Get full credit. So this is showing you the three different ways that you can get rid of those acetylcholine molecules or other neurotransmitters. So we saw them coming across, we saw them binding, but you can either enzymatically break them down, you can either recycle them into a glial cell or right back into the presynaptic cell. Both of these are ways to recycle them. And then the last way is into the blood, where it goes to a different part of the body, is broken down and then sent back to neurons to work. So this is the second part of the essay, asking how does the signal spread through the synaptic cleft? How does this work? Okay, that is it for today. I'm sorry none of you are able to be here, but I would have liked to change the time, but I didn't have time any other time. So I hope to see you all in lab tomorrow. I hope this helps you, and see you tomorrow, and enjoy the rest of your week. Bye, guys.